Thank you, דוקטור שניידר, עבור ההרצאה. החברה השנייה העוסקת בטיפול גנטי היא חברת פייזר. את מצב הניסוי הקליני שמתבצע בחברת פייזר יציג דוקטור דמיאן סימונאו. דוקטור סימונאו סיים לימודי רפואה בפריז ועבד במשך כ-18 שנה בחברות תרופות שעוסקות בין השאר בפיתוח טיפולים למחלות נדירות. כיום הוא עובד בחברת פייזר כמנהל בכיר בתחום מחלות נדירות בשווקים מתפתחים. דוקטור סימונאו, please.
Good morning. I would like, first of all, to thank you all for being here today, for inviting us. It's a real honor. And I would also want to particularly thank the organizers for the very warm welcome we had in Israel yesterday. Thank you very much. I would like to start by providing an update on one of our programs on DMD you might have heard about, which is domagrozumab. So domagrozumab is a monoclonal antibody that was designed with the idea that it would act against myostatin. As you might know, myostatin is a protein that inhibits the muscle formation. So basically by inhibiting the inhibitor, the idea was that we could favor increasing the muscle mass, which we have seen already in animals, for example, in animal models. And we were hoping that that effect was going to become functional also in humans. So with that in mind, we went up to a phase 2 study, which unfortunately had to resume in August, on August 30 last year. As you can see here, however, we managed to include 80 patients in the active arm and 40 in the placebo arm. And as you will see, most of the patients also, as you can see on the bottom of the slide, included were between 6 and 10 years. And as I mentioned, the study did not meet its primary efficacy endpoint. We did multiple analysis in trying to detect any signal for efficacy. However, these were not confirmed. And the totality of evidence of the functional endpoints, muscular endpoints, did also not contribute to the overall treatment effect that we were hoping. However, the good thing is that the studies were not terminated for reasons of safety. We still are looking further exploratory in analysis to understand whether potential benefits could be still identified. And despite disappointment, the study data sets will benefit for future studies because we have very important information which was generated, especially in the placebo arm, which allows me to transition to our DMD gene therapy program, because patients studied on placebo contributed to our knowledge on how to best design clinical trials for gene therapy. We are definitely grateful for the families and the boys who contributed to the study. We are grateful also to the investigators. I would like to mention that we did not have any sites in Israel for that program. We also, of course, we're grateful to the patient advocacy group, and we look forward to tell you more when we'll have more results. And with that, I would like to move to the most exciting part of the morning, which is our mini-dystrophin gene therapy program with our compound Pfizer 9926. Just to recap a little bit for those of you who are a little bit more familiar, as you already heard, gene therapy is a general strategy used to replace or repair a dysfunctional gene. Most widely used for diseases caused by a single gene mutation, of course, in an approach to improve cisment, or, which is our case in DMD, correct a disease towards health. You heard already about the vector, which is an important part of the construct of the gene therapy. also called capsid, which is the envelope of a non-replicating virus, delivered to a particular what we call target, which is a part of the body or part of the tissue. The currently used vectors are AAVs. Ours is AAV number 9. I will come back on that. And you can see here, in orange, you can see that the therapeutic gene has to be included in that vector together with a promoter. The promoter being the genetic part, which helps the expression of the gene in the right tissue. The issue is, as you might already know, the limited capacity of the capsid to host a sizable gene and a promoter. So here, again, you can see on the top, the size of the gene encoding for dystrophin is very large and very big compared to the carrying capacity of the capsid. Therefore, as a general approach, the AAV gene therapy requires miniaturization of the DMD gene, and this is common across all the different programs that you already heard and you will continue hearing in the morning. It requires the delivery to muscles, as you know very well. So basically, the gene has to be expressed. There it counts in boys, in muscle. And as you know very well, muscles represent a very important part of the total body weight, which we estimate around 40%. Therefore, unlike the eye, for example, where approaches of gene therapy required a certain amount of vector being produced, for the muscle, you need enhanced, a much larger quantity of product in order to be able to start studies in humans, especially when you start looking at higher doses. The serotypes with a selected tropism for skeletal and cardiac muscles are very important, and therefore our AAV number 9 was studied in order to be able to fulfill this criteria. Also, the entire part of immunology and immune responses directed against the capsid, AAV, or against the antidystrophin, the minidystrophin itself, is very important to be studied before studying in humans. And a lot of effort has been done by our researchers in that area. Also, the levels of miniaturized dystrophin expression required for improved functions have also been studied before going to humans, because as you know very well, in gene therapy, we have one chance to make it right. Once the infusion is already done, it's too late to come with a second dose or another approach. Our program has a potential, also therapeutic potential for all mutation types. I will not come into details into the gene therapy timelines. Basically, it started in the 80s with the identification of dystrophin, the first use of AAV as a vector, and very recently in the 2018-19, the initiation of the first systemic AAV truncated smaller dystrophin trials began. So with that, I will move to our Pfizer 9926 background and rationale. We call it minidystrophin. Basically, there is no difference with other approaches called microdystrophin, for your knowledge. It's a sequence based on a mild Becker muscular dystrophy patient's gene sequence. I already talked about our serotype 9 AAV9 AAV that we selected, which is non-pathogenic, and has a high affinity for skeletal and cardiac muscles. We also have a muscle-specific promoter, as you have seen before, and you can see in the red pictures downstairs, the expression has been confirmed in animal models and is it's done at relevant levels. You can see that in the green bar there, this is our dystrophin mini gene with the promoter that is associated with it. So this is basically the biochemical construct. And with that, after a thorough analysis in animal models, we decided last year to move in humans with the first administration in March 2018 in humans, in boys, with the first phase 1b study. And one important concept here is that in order to try to make boys' life as easy as possible, of course, within the constrained environment of clinical trials in this area, we consulted a lot with the patient advocacy groups, and moving forward, it's our general requirement that as much as possible, we would like to hear from you what matters to patients when they go into clinical trials. There are so many things that come from administrative aspects, administration times, etc. So here is the design of our phase 1b study. It is a safety study. As you can see, we include patients from 5 to 12 years old. They have to be ambulant and on daily glucocorticoids. They do not have to have antibodies against AAV9, obviously. The study consists of a single intravenous infusion, and we have a staggered dosing approach. What does it mean? It means that we have a sequential approach which allows us to apply any learnings that we have from the first patients that are dosed, if there are any, especially in terms of safety, to the following group of patients when they are administered, and not to start all the patients from the beginning. You can see on the upper part of the slide that we had already biopsies at baseline, also at month 2, interestingly also at month 12. We also have a long-term follow-up up to 4 years, so the study will not stop after month 12, but will continue in order to continue studying, especially the safety aspects. So where are we with the study now? The study is conducted in the United States. We have 3 sites activated. 11 boys were recruited. 3 went in the process of screening failed. 2 were pending eligibility as of February 2019. And overall, we have 6 patients exposed to the treatment, both to the lower dose and to the higher dose. And you can see here the exposure period, patient by patient, with the longer, with the patient that had been followed up for the longest time, being around 12 months, studied for 12 months, around 12 months now. So as a summary, Pfizer 9926 is an AAV mini dystrophin with a specific vector, which is important. The current clinical phase 1b opportunity will allow us to learn more about short-term and long-term safety, obviously about the level of mini dystrophin expressed over time. We will also look at the relationships between levels of expression and biomarkers, MRI muscle, and clinical function. We'll definitely study AAV immunology and dystrophin immunology. The initial data out of the 6 patients will be presented end of June 2019. And we have a very, last but not least, as you already heard, in that area, both for the conduct of the clinical studies, but even more importantly, when these products will become available, what will be very important will be the ability of the manufacturer to produce the drug. And with that in mind, we really invested in a high-scale manufacturing capacity for gene therapy. As in Pfizer, we have gene therapy studied not only for Duchenne, but also for several other diseases, for example, hemophilia, but many others to come. So with that, I would like to ask if you have any questions. Okay, so thank you very much then. Oh. Sure. I need to be very careful because, as you know, these data are late breaking. We are still analyzing them. Not all the data is for the moment there, so it's work in progress. We have high hopes that we will be able to present them at PPMD conference. I will come on that. You will have it here. Yes. I'm not 100% sure about the target for recruitment, but what with the follow-up of 4 years, you can estimate an overall duration of trial around 6 years at least. Yeah. I will have to check that out. I will be very honest with you. Yeah. So this is the elephant in the room, right? So yes, we are planning a big phase 3 international trial with sites outside of the United States. We are in the process of selecting the countries and the sites, and we hope to be able to come back to you with good news. However, of course, there are so many aspects that impact the ability of a center to be included in our program. But this is work ongoing. Sure. It's very, very hard to say. It's very, very hard to say. I fully understand the urgency because you face difficult personal situations and family situations. But you also have to understand in the area of gene therapy, we don't have any analog. And we learn also one from each other, program by program. What happens in one help us improve the other, whatever the company is. And I think the point was already made by my colleague in Solid. So it's very, very hard to predict a duration of availability. However, what we can assure is that we do our best to accelerate the programs, and as I mentioned, to be able on day D to be able to produce enough amount of drug for the commercial needs. Another question. What is it now? Because we know the virus is for many years around. The virus that can take. What is it now that brings so many companies working on many disciplines? Is it the manufacturing process that is more available for companies, or what is it new in the technology that enables companies like yourself So I only can comment for Pfizer, obviously. There have been significant improvement in the ability to produce. So basically, a study like the phase 3 that we're planning, that I already mentioned, would have been unconceivable only 3 years ago. And there has been significant improvement in the manufacture of both techniques, scalability, learnings, because, of course, you have to produce a lot, but another key aspect that is crucial is the purity of the compound when dealing with DNA. And this is something that we have been very thoroughly looking in order to avoid any accident.
Why would you say that it's possible only to use it once?
With the current products being tested, okay, in the future. Why? Because for immunity reasons, once you administer once, there is very likely that you will have antibodies against the second dose. That will, and of course, this is something that you want to avoid. With the current generation of AAVs, we don't foresee a lot of ability to administer a different drug because we suspect that the antibodies will be also acting against the different products. However, we also have hope that new generations of capsids will be produced, but we have to, again, we're going into the unknown, so we have to start with what we know. So for the moment, each company has its own capsid, and we have to start from somewhere, just to keep it going. Yeah.
Or are you hoping that you could cure the disease, or there's nothing like that that is being expected?
With the mini-dystrophin we call it mini, so with the mini-dystrophin approach, our ultimate goal is to replace it with Becker indeed. Again, other types of capsids, other types of viral vehicle in the future, because we're limited by that size of capsid. We cannot put the whole gene. In the future, maybe different types of vehicle will be created that will allow the expression of the full gene, hopefully. Thank you very much.